Welcome everyone, and let me give some overview about my research group's work in this topic. So first of all, I am really grateful to the grant agencies that are supporting our work. So one is important National Laboratory for Health Security in Hungary. I am happy to chair the Pragmatic Coast Action, which is actually a very important network of scientists, over 260 scientists now, working on ticks and tick-borne pathogens, and of course the PREPARE-TID network for the rapid diagnostics. I am leading the research group for emerging pathogen ecology, which includes both senior scientists like Daniel Brooks and postdocs, PhD students and master students, and all of us are working on the eco-epidemiology of ticks and tick-borne pathogens and of emerging pathogens in general. So I would like to start today with the so-called parasite paradox, which means that we all know that pathogens are considered ecological specialists, which means that they are strongly co-adapted to their hosts. But we also experience that emerging infectious diseases occur very rapidly, and the shifts from the unrelated hosts are very common. I would like to explain why this paradox exists. The basis for that is that we have a wrong so-called classical coevolutionary model. This model states that within natural hosts, there needs to be a new mutation, a genetic change, in order to colonize a new host, like a bat virus colonizing a human. In this situation, this new mutation is usually quite rare, and for this reason, emerging infectious disease events should be also rare. And the other consequence from this theory is that it should be unpredictable. So the good news is that it is usually not unpredictable. The bad news is that it is not rare. Why is that? It is because actually this classical coevolutionary hypothesis is not working. It is not the real time, the real situation in nature. So we need another paradigm, and this is called the Stockholm Paradigm. The Stockholm Paradigm was developed by Daniel Brooks and colleagues over the last 2 decades, and the basic statement of this paradigm is that within the natural hosts, there is a huge natural variability, both genetic and phenetic variability of several pathogens, and some of these variants are able to colonize new hosts if there is an opportunity for this. And this is the case which we have been experiencing during the COVID pandemics. So from a natural occurring virus, there were variants which were able to colonize human hosts and also domestic dogs, for example, or deer or rodents. And because these variants were different, they were able to also find different hosts to colonize. The only pre-existing trait was that there should be a contact between these hosts. So there has to be an opportunity. And if within this host, of course, these new variants will modify genetically and will be able to spread even at a better rate compared to the original pathogen. So actually, this paradigm was developed before COVID. So this book about Stockholm Paradigm appeared in 2019, and it's based on ecological fitting, a long-established phenomenon, which says that there is no need for mutation or genetic change for host colonization. And this also means that with climate change or habitat loss, there will be an increase of the migration within vectors, hosts, and pathogens. And this will lead to new contexts, new opportunities for emerging infectious diseases. So with other words, we can say that emerging infectious diseases are the rule and not the exception. If you consider global environmental perturbation like climate change, which is not only global warming, but as you know, extreme climate events, like, for example, the last year's terrible disaster in Valencia, which took over 130 lives, these kind of changes were described to help disease vectors, pathogens spread at a higher rate. This paper from 2020 shows how strong the connections between variable climate change factors, like here on the left side, warming, precipitation, floods, drought, and so on, are connected with various types of disease. So, for example, as you can see, vector-borne diseases are the most affected by climate change, and this enables mostly viruses, but also bacteria, fungi, and protozoans to spread at a higher rate. If you add natural biodiversity loss, which is very common, unfortunately, on the global scale, it is also leading to new contacts, because those hosts, including humans, which were never able to meet parasites and pathogens within the natural habitats, are now able to have a contact, and this is the way how new outbreaks and even new pandemics can develop. I would like to add another problem, which is our very scarce knowledge about the microbes around us. To give an example, just among the viruses found in birds and mammals, we know only a couple of thousands. For some of them, we only know the name. However, according to the current estimates, there are 1.5 million of these bird and mammal viruses. Among them, 500,000 is able to infect humans. So this is another huge problem, that we haven't described the diversity of microbes around us. If you want to consider the money side, we can calculate easily that BC, so before COVID, the money spent yearly for EIDs was approximately 1.3 trillion US dollars, and only a single EID event, COVID-19, has cost us already several 10 trillions of US dollars. So it's easy to say that prevention would be much more a logical step compared to the answers or current situation that we are answering to the situations just later on and not before that. A further problem is urbanization, which is an ongoing trend on the global scale. More than 55% of the global population now lives in big cities, and it is estimated that very soon it will be 2/3. And a problem with cities is that those green spaces, which are otherwise necessary for human recreation, are also perfect places, for example, for rodents, for songbirds, which are very good maintenance hosts for blood-sucking parasites like ticks and mosquitoes, and also for tick-borne pathogens themselves. So these can be also actually hotspots for some of the vector-borne diseases. If you consider the example of Southeast Asian big cities where wet markets are also common, especially after the SARS epidemics and before the COVID epidemic and pandemics, again, lots of wet markets opened and reopened. So this is actually the perfect storm for disease emergence, because we have lots of dead animals, lots of live animals together with humans in a very small vicinity. And adding globalization, we have actually every step necessary for developing a new disease case. Just a figure from the best pandemics report, 24% of wild terrestrial vertebrate species are traded globally, which again is a very high risk factor, very important risk factor for disease emergence. So with a metaphor, we can say that we live in a minefield of evolutionary accidents waiting to happen. And this is not just a metaphor, because as all of you know, although this is an 8 years old graph, we can put many, many names on the global map where different viruses, bacteria, fungi have been emerging. And this trend continues to be the case in the future. So after this lots of bad news, my question is, or our general question could be, what can we do? And this is what I would like to talk a bit more today. The good news is that we can be proactive about coping with the emerging infectious diseases based on the Stockholm paradigm, because colonization of a new host requires new opportunities, and specific pre-existing capacities are given. So these capacities are actually predictable, and this allows us to anticipate disease emergence. So we can be proactive instead of being reactive as in most cases before. And the answer is actually the DAMA protocol. DAMA stands for 4 letters: documentation, assessment, monitor, and act. And to put it very simply, DAMA protocol is about finding the pathogens before they find us. It has been published over 10 years ago by Daniel Brooks and colleagues and summarized in this 2019 book that I was showing. So very simply, we have to document pathogens, microbes around us. However, we cannot document everything, so we have to assess the threat that they are posing. And if they are found important or dangerous, we have to go back and monitor their spatial and temporal distribution, the changes in the populations of hosts, vectors, and pathogens. And maybe the most important fourth step is the action. So we scientists are in general the first three steps. However, the last step is lacking in most of the research proposals, grants, and projects. Of course, we scientists are not able to do the actions against the pandemics or epidemics themselves. However, we can provide information for the stakeholders, for the decision makers to act proactively instead of react to the situations. To give some examples, documentation can happen very easily by microscope, by mobile phones, and observations, simple observations. It is important because without documentation, we won't be able to find the disease X, which, for example, the PREPARE-TID project is also looking for to monitor and find, because COVID-19 used to be a disease X before. Now we have the name and we have many experience with that, but there will be new disease X agents. And on this WHO priority disease list, you can see COVID-19, Crimean Congo hemorrhagic fever, Ebola, and so on. So these are all zoonotic diseases. And of course, I have to also emphasize that we have to understand the ecology of the hosts and of the pathogens themselves in the natural cycle. Concerning tick-borne diseases, the easiest way to document things around us is to go out and flag for ticks. Flagging will collect the ticks in the natural environment, and we can quantify them and we can check them for pathogens. And this leads us to the second step, assessment, because if we want to assess the risk of ticks around us, we have to first of all identify them and then screen them for pathogens that they transmit. Then there is the so-called phylogenetic triage. This is the most important tool for the assessment. Very simply put, we have to put the question, is this a known pathogen or is this closely related to a known pathogen? If the answer is no to both questions, we can ignore it, but it's good to archive this pathogen. If the answer is yes to either of these questions, then we have to gather more information about its transmission dynamics, microhabitat preferences, and natural history. The third step is crucial. Usually, we can think in 4 years research grants, and the huge problem with this short-sighted vision of grants is that there is no money usually for monitoring. However, monitoring is really crucial, because if we cannot see the temporal changes, for example, in a host population or a vector population, we are not able to prepare for the next steps. And the next step is action. Action is actually cooperating and coping with the problem. Example, we have to teach citizen scientists how to reduce the chance for disease establishment. We have to reduce the risk exposure, recognize new arrivals, and mobilize universities, NGOs, governmental agencies, and so on. We have to provide proactive suggestions to the decision makers in order to prevent the next pandemic and not to respond only to that. And international scientific cooperation is actually one of the best tools to do an action. I would like to give you also an example of what my research group has done in the case of, as an example for an action in the DAMA protocol. So the next couple of minutes, I will talk about citizen science and its usage in ticks and tick-borne pathogen science. As you well know, citizen science is a perfect tool if there is only a few scientists, which is usually the case, and scientists only have 2 hands in general, but there are lots of helping hands out there who want to help. We just have to tell how and what to do. For this reason, we have started a citizen science project about hyaloma ticks. Hyaloma ticks are Mediterranean and African, Asian tick species, which are quite common in tropical and Mediterranean countries, but they are not present in Central and Northern Europe. They are so-called two-host ticks, which means that the first host, usually a rodent or a bird, will feed the larvae. The larvae will molt on the host and stay on the host as nymphal stage. They will also feed and feed for long, elongated times, and will only drop off after they have completed the blood meal. If it is a migrating bird, it can spend several weeks on the bird, which means that it can take several thousands of kilometers and be taken, for example, from Africa to Central Europe. If it drops off from the host in Europe, it will molt to the adult stage, and it will look for the second host, which is an ungulate, horse, or human being. Here you can see the bird host infested with ticks, and every year several millions of ticks arrive in Central and Northern Europe. And with climate change, there are more and more cases of adult observation of adult hyaloma ticks, because they find similar circumstances in Central Europe compared to the Mediterranean region. Why is this tick species so important? Because this is the main vector of Crimean Congo hemorrhagic fever virus. This is an RNA virus and has around 30% case fatality rate. And unfortunately, currently there is no vaccine and no specific treatment against this virus. So this is why we have to monitor its presence. So this is the reason why we started this website, both in Hungarian and English. It is called the Tick Watcher in English, and this is about finding these large and quickly moving ticks, which have stripes on the legs. So fortunately, they are actually very obvious, and by naked eyes, even lay people can differentiate them from Ixodes ricinus, the common sheep tick, everywhere found in Europe, in the middle, and for example, Dermacentor ticks, which are common on dogs in Central Europe. We put the main, most important information on the website with photos and even with videos. We explained how it looks like, and we asked people to send in photos and even the specimens in a zip bag or in vials. Here to the left, you can see hyaloma specimens on a shoe, and to the right, you can see the video how quickly they can move. So this is actually our luck, because they are very different from the tick species found all over Central Europe. They are running very quickly. They are not waiting for the host, but they are so-called hunting ticks, which will run for several hundred meters even, find the host and attach for taking the blood meal. This video was taken, by the way, in Turkey, where Dr. Vatansever hosted us for a training school for hyaloma and CCHF studies. Amazing ticks, by the way. So if you see such a tick, you won't forget it. If you produce a website, it is not enough, because people won't know about it. So we went out to public events, for example, to the researchers' nights, to talk to people, to show them what ticks are, why are they dangerous, what do they have to take care about. We even designed with my PhD student a tick escape room, where with several tasks lead to solve the problem. And here you can see that many children were interested, maybe sometimes frightened from the plastic ticks that we showed them. By the way, it's her mother, not me, frightening with the tick. So it is very important to talk to people and to let them know that we exist. And soon people started to send in pictures. As you can see, they send bugs and beetles and spiders and every kind of biodiversity. But hopefully, or luckily, we also received many pictures of ticks, several hundreds per year. And it is also very important what you show to the media, because, for example, in this Hungarian newsletter, you can see that another tick species was shown, not hyaloma with the striped legs, but Dermacentor ticks. And soon we received many Dermacentor ticks. The good news is that actually we can make use of this bycatch as well, because with this we're able to map other tick species in Hungary as well. This is, for example, a soft tick, which is not very common in Hungary, but it has been found within the city central of Budapest in an apartment. Or this one was sent by a mother from her daughter, showing a tick in the eyelid. And sometimes not the best or nicest pictures are the most important ones. Also here you can see it's quite blurry picture, but you can see the stripes here on the legs, which show that this is a hyaloma tick. And indeed, this was removed from a cattle, and we could identify this as a hyaloma rufipes. We also did the genetic analysis based on the mitochondrial DNA, and we could show that this is belonging to the European African clade of this tick species. So probably it was taken from Africa to here. And we also received samples from dogs. For example, this one is a hyaloma marginatum from an adult, from a dog and we also screened this for the CO1 gene, and you can see that this one belonged to the Eurasian clade, which means that this might have been taken from the east, from another migratory bird route. And we received samples from donkeys, from kitchen floors, even during lawn mowing. Some people found within Budapest, within the city, hyaloma ticks. So it was it is quite a success. We published the first year's results here, and you can see that the bycatch itself was very interesting, because we could see that several other tick species were also sent in from different corners of the country. Here you can see the summary, what tick species or how many hyaloma specimens were found during this 5-year project so far. Until today, 26 specimens were documented. And interestingly, as you can see, in some cases, like here or here in the south, there are some accumulated cases, which means that not only in 1 year, but in several years, different specimens were found on the same location. And this is probably because a new local population is developing here. So according to our current knowledge, with these very mild autumns and very mild winters that we have nowadays, it is very probable that we will soon have a local hyaloma population within Hungary. We also screened these ticks for Crimean Congo hemorrhagic fever virus with the help of the BSL-4 laboratory of the National Laboratory for Virology in Pécs, in south of Hungary. All of the specimens screened so far were negative for CCHF virus, but we have also performed the metagenomic analysis and found a very interesting related virus. Here you can see the big group where Bunya viruses belong to. And within this, the group itself is now called Bunya varicetes, and within this group we found a distinct clade, and on this distinct clade we detected a single, in single case, a virus called Wolt's tick virus, which is very closely related to Bunya virus-like viruses detected in Croatia and Norway. So further research has to clarify whether this is a pathogenic strain for humans, whether this is a tick-specific virus, or it can be found in vertebrate hosts as well. So several questions have been raised. However, we were able to characterize the whole genome of this virus, and we published it recently in scientific reports. So to summarize, I would like to quote one of the sentences of Daniel Brooks and colleagues' books, for a book from 2019. Our protocols must be evolvable. There is no static solution for problems involving an evolving Earth and an evolving biosphere. So this means that it is not enough to document, assess, monitor, and act. We have to recycle this process, and we have to go back and find out what has been changing in the situation. For example, in the presence of hyaloma ticks, which part of the country are those ones where local population will try to establish. We have to also get in contact with the public health authorities, with the veterinary health authorities to cooperate. And of course, we also have to improve our methods. One of these improvements is the latest that we tried is a mobile application, which is now available in Hungarian and in English, and is currently working in Hungary. This is called a pragmatic mobile application, and with the help of these, people can send in photos and report hyaloma and other tick observations in a faster way, and we hope that it will enhance our efforts to prevent the next disease outbreaks and to monitor the presence and occurrence of both ticks and the tick-borne pathogens. So our hope is now that we have a good chance to translate this to several European languages. Currently 17 different languages are translated to. So hopefully within the beginning of next year, we will launch this application in several European countries. So to summarize, my main message for today would be that let's contribute to this DAMA protocol more and more, and let's hope that people and scientists and decision makers will be also knowledgeable and try to do the best to be proactive. Thank you very much for your attention.
vector-borne pathogens. So let's start with a few definitions to be sure that we all talk about the same things. So vector-borne pathogen are spread by vectors, which are hematophagous, can be mosquito, ticks, sandflies, and so on. Most of them can cause zoonoses, which are diseases that affect both animals and humans. And so vector-borne pathogen can be parasite, bacteria, or viruses. According to World Health Organization, neglected disease origin from tropical or subtropical region and affect mainly low middle income country, but are also spreading worldwide. And emerging disease, there are diseases that come for the first time, or also can be diseases that have a repeat increase of the incidence, or that have repeat increase of the geographic range. So what is important for those diseases is to have a rapid detection to prevent their dispersion and also worsening symptoms in case of human and animals infected. I try to sum up the workflow to detect vector-borne pathogen. Let's start with collection samples. You will have also the storage and the cleaning steps within. After, you need to prepare your sample. It can be derivation of lasing of it. It can be also the inactivation of it to handle it safely. Usually you have a step of nucleic acid extraction, and then you will detect your pathogen of interest. Finally, you analyze your result and conclude if you have or not your vector-borne pathogen in your sample or not. I will take the example of ticks because that's what I'm working on in the daily life. And for ticks, it's quite a challenging sample type. You have soft ticks and hard ticks, and they are varying in size. You have very small larvae, you go to nymph and adults, and as they are hematophagous, they took a blood meal, and it took it a few days to almost 1 week or more. And so they get very large when they are engorged compared to the size when they are unfed. And also, what is interesting to note is the blood act as an inhibitor in many detection methods. So engorged ticks are differently handled than unfed ticks. To collect your ticks, you can collect the host-seeking ticks on vegetation, with flagging, for example. For soft ticks, you can look for them in nest or in a burrow, or you can collect the engorged ticks directly on host. For the storage, I would say the best way is to collect them and bring them back in the lab fresh. But sometimes it's not possible, or you have to send them away. So you can use, you can store them frozen and send them in dry ice. You can use RNA later or store them in ethanol. Those steps, storage and cleaning, are important no matter what kind of sample you are handling, but especially for field-collected ticks, it's important to note them because it will force you into a way or another of the extraction methods and the detection methods. So for the description, you need to break the hard tegment of our ticks. You can use chemical way like proteinase K, but the main way is to physically disrupt them, like cutting them in half, the manual braiding, or also the use of bits beating. After that, you will go for DNA or RNA or both extraction, which depend also on how you store and collect your ticks, and also which method of extraction you will use. Also, what is important to note is that all tick-borne viruses except African swine fever are RNA viruses. So if you want to detect them, you should go for RNA extraction, and so proper storage condition. So depending of your research question, you will either know which pathogen you are targeting, and you go for a targeted method, or you don't really know which pathogen you want to see, or maybe you are looking for different pathogen than usual. So you need a method without a priori. So I will start with the targeted method. So the gold standard is the polymerase chain reaction, PCR. You have all different PCR, quantitative one, nested, RT-PCR. You can also have multiplex when you're targeted multiple gene. So PCR are conventional at host friendly and also specific. You use different primer, and you can either choose to target the whole genus, for example, the whole bacteria, Borrelia. So we get all the bacteria within your ticks. You can also have primer for a group of pathogens. So within Borrelia, you have, for example, the Borrelia that cause Lyme disease. You have also the relapsing fever Borrelia, and maybe you will only target the Lyme disease one. So you have like a 20-ish species of Borrelia targeted by the same primers. Or you also have a specific primer for only one species within all the Borrelia possible. So for example, a primer for Borrelia garini or for Borrelia azedi. So depending on the primer you are using, sometimes it's quite challenging to detect co-infection. Co-infection by the species from the same genus, for example, but also if you don't look for other pathogens, you will miss co-infection between two different tick-borne pathogens. But it's the most widely used technique to detect tick-borne pathogen within ticks. Another way to detect your pathogen is the use of microfluidic chips. So it allows you to detect a large amount of different vector-borne pathogen and run many samples at the same time. The main constraint is the cost of the chips and also the design of the primer used in it, but it's used for tick-borne pathogen detection. We have the loop-mediated isothermal amplification called LAMP. So think about a PCR, but made at lower temperature and also with specific primer that make this loop that you can see. So because you are running at lower temperature, it reduces your reaction time, so you get your result faster. But the main constraint is the specific design of this primer that should make some loop during the amplification step. So for tick-borne pathogen, it was successfully used for detection of Rickettsia and Borrelia bacteria within ticks. You have also the recombinase polymerase amplification, RPA. So again, it looks like a PCR, but it's using a different primer that interact with some proteins. It's working at lower temperature between 37 and 42 degrees usually, and you will have your result in only 20 to 30 minutes compared to PCR, which are likely 2 hours or more. Again, the specific design of the primer can be challenging because it's not available for all tick-borne pathogen, and so not all vector-borne pathogen. And for ticks, they only succeeded yet from tick-borne pathogen that were from culture, but not directly on tick samples. When you are looking for an emerging pathogen or for a new one, you need a method without a priori. So I will only talk about next generation sequencing, but there is other techniques. What is useful is, according to your library you use, you can detect all the pathogen. We'll know everything about it. There is strain, and you can have a lot of the genome in your sample. But the analysis is challenging and require bioinformatics skills because you will have also data from your sample. So if you are looking in ticks, you will have the ticks genome, you will have the microbiota that you need to look and distinguish between what is corresponding to your tick, to your microbiota, but what is also looking for your pathogen. And the use is increasing in tick-borne pathogen detection. I won't go further into this method, but you also can use microscopy, for example, to detect parasites within the blood smear. You have DNA hybridization, capture sequencing assay, or even CRISPR-Cas9 that can be used for nucleic acid detection of your vector-borne pathogen. And if you are looking for proteins, you can even use Maditov mass spectrometry to detect if your sample is infected or not by your targeted tick-borne pathogen. We'll make a short about lab suite case because the goal is to have a mini lab directly on the field using robust and rapid method for extraction and detection of your pathogen. So let's think about you have an emerging disease somewhere in the world and you don't know what is it yet. You go there and you collect like every sample that you could. Environmental one, potential vector, animals and human samples like blood or serum, and you can extract it directly on the field. You will have a lab a suite case with everything you need from the lab. You do the extraction, so you need to be quite rapid. And then you will detect your targeted pathogen. So usually it's by RPA, lamp, or loop. So the isothermal amplification, because in the field, you can have limited electricity, limited space, and you shouldn't be the whole day on the field because of the weather or the political situation, for example. That's what we are doing it in the PREPARE-TID consortium. So currently under testing, we have manual grading of ticks. So we tested various species, namely Ixodes ricinus and Dermacentor reticulatus, different stage, so nymph, adult, male, female, and also different fed status, so non-engorged and engorged tick. So the protocol is having both physical and chemical leasing with a heat inactivation step. And we will extract our nucleic acid by a method called reverse purification, So both RNA and DNA are preserved, which is very, very good because for tick-borne viruses, they are mainly RNA viruses. And we compare three methods. The first one is what we do usually in the lab. So bits bitching for the grinding and RNA extraction looking with a manufacturer kit. We have the manual grading and the reverse purification from Expedit. And we have a third method using the bits bitching, but with the reverse purification extraction. We did it with Ixodes ricinus that were naturally infected by Borrelia. So all methods allow you to get nucleic acids. We run some PCR from both of the tick 16S and for the pathogen, so the Borrelia bacteria. And we also run them in qPCR. And what is interesting, we have correspondence between PCR and qPCR results. And the CT value are quite the same. We also did it with another vector pathogen system. So with Anopheles, I'm pretty sure it was Anopheles Ecology, that were artificially fed with blood infected by Plasmodium falciparum, with a mean prevalence of 70%. And in this case, both method 1 and 2 gave results, but the latest method didn't work for the detection of the pathogen. So to sum up, it's a challenge to detect neglected and emergent vector-borne pathogen because you have various sample type, so you have various methods of detection that can be directly on the field. So depending on the question you are looking for, you need to think about how you will collect your sample, how you will preserve them, and how you will detect the pathogen of interest you are looking for. And also what is interesting is the still use sequencing for, of course, unknown pathogen, but also to look back in old samples to check if a pathogen that weren't of interest yet was actually already in the sample type. Thank you very much.
I will focus mainly on my work in the field, and it will involve Crimean Congo hemorrhagic fever and ticks. But let's start with ticks and tick-borne surveillance in Turkey. Well, I'm sorry to say that there is no national surveillance program in Turkey. That is based on the human cases, and that's all. There is no epidemiological, no ecological work on diseases and ticks. There is only one passive surveillance I remember. It was in 2008, and at that time we recorded about 300,000 tick bites on humans that year. That's all what Ministry of Health did for surveillance of ticks and tick-borne diseases. There is National Zoonotic Disease Action Plan actually, but there are objectives for surveillance, but there is no action because of limited funds, I will say. There are some works, very small scattered works of surveillance and ticks and tick-borne diseases, but they are not.
Sorry, I don't want to interrupt you, but some people in the audience said that the slides are very blurry. Can I ask you maybe to turn off the camera maybe and just maybe the slides because maybe it's a problem of internet.
Okay, how is it going now?
I see them a bit better. I think it's for you guys. I think it's still a bit blurry, but it's a bit better.
Oh, probably the internet. Yeah.
Maybe it could be the signal. Yeah.
Well, I have a good signal here, but sorry for that.
No worries. Maybe let's continue maybe because I can still read. Slightly better. Okay. And we can maybe continue, and then if it's still very very blurry, maybe we can stop again. But it's good like this.
Yeah. Okay. I can start the problem. Okay. And there's very small number of work done for ticks and tick-borne diseases, but they are mostly for publication purposes and they are not systematic. Well, I will concentrate on the work which is done in the field regarding CCHF and ticks, vector ticks. Well, the Crimean Congo hemorrhagic fever disease is widespread in Turkey. It's the largest epidemic in the world.
Sorry, I don't want to interrupt you again, but we lost the presentation now.
Oh, really?
You're not sharing, yeah. Sorry.
Oh, okay. I'm going to share again. Can you see or not?
Yes, yes, a bit blurry, but it's okay. We can continue. I think also for the audience, it's okay. But we can share the slides afterwards. Let's say at the end of the talk,
I will
we
do
can.
it.
If you someone in the slide, maybe we can share them via email.
Yeah, I will do it. No problem.
But it's still off. It's still not sharing.
Oh. Can you see it?
That's perfect. Okay. Nice, perfect. Nice, perfectly defined. Okay. Okay.
Okay. Human cases. Oh, it's not working. Okay.
No, it's okay. It's okay. It's okay.
Okay. And the vector of CCHF in Turkey is yellow marginatum. So it's two-host tick, has a 1 year of life cycle from egg to egg, and adults feed on artiodactyl animals like cattle, horses, and wild boars. They don't like much the sheep, and they peak activities between May and June. Immature larvae and nymphs, they prefer small wildlife, like mostly hares in Turkey and sometimes hedgehogs, and ground-feeding birds. In case of Turkey, this is partridges, but in case of some Russian part of in some Russian territories, it could be a rooks as well. It's a hunter tick, aggressively attacks humans. It moves very fast, and but it doesn't climb to vegetation, so we can't collect it with dragging or flagging. There are some accounts about yellow mallustanicum in Spain, which is collected by flagging as well. But yellow marginatum, it's hunter tick and doesn't climb to vegetation. It's active in summer when temperatures reach 10 degrees. And during the hot days, they are very active. And when there is a too hot, they hide under vegetation in cracks, and they preserve the water balance. They overwinter as unfed adults in cattle pastures, in fallow lands, in bush, but not in livestock shelters. So the place we can find yellow marginatum is in Finland. Well, if you want to do a targeted surveillance about CCHF and ticks, vector ticks, you need to know something about the ecology of the tick, and in the same time the life cycle of the ecology of hosts as well, like cattle and hare in Turkey. And on the other hand, you need to know the land use and habitat in area where you're going to sample to do surveillance. It's very important. They get active in summer, and the adults go to mostly to cattle. They prefer cattle, and they don't go to sheep much. And they feed on cattle. If they are infected, they can transfer the infection to cattle. If the cattle is infected, they can accrue the infection from cattle. And they feed for 9 to 14 days on cattle and drop as engorged female. And at this stage, there is transmission of the virus from infected female to the larvae. And those larvae, in the end of June, beginning of July, they start to go to hares or ground-feeding birds in the field. They feed on them for about 2 weeks and drop us engorged nymph. Because this happens to the end of August, from the mid of August to the end of August, And during that time, the days are shortened and the temperature starts to drop. And the tick molts to adult, but doesn't go to search for another host, and it's going to hibernate. After September, we don't see Hyaloma ticks in the nature. Well, if you intend to do surveillance about ticks, targeted tick surveillance, we can focus on host-seeking ticks. It's very good indicator of prevalence and can be used. The data can be used directed for human risk estimations. You need to know we need to have knowledge of habitat and land use in the area you intend to work. You need to use right method for collecting ticks. You know, for humans, for Hyaloma ticks, the dragging or flagging doesn't work. You need to go to the field and stay and let ticks find you. You can, you can use vibrations, CO2, and they have a good vision and they can sense you and run up to you from meters away. And when it comes to testing of unfed ticks for pathogens, it's important when you collect the ticks to maintain them. The maintaining conditions are very important. I usually prefer to transfer them alive to the lab, identify them, and then store them at -80. You can use RNA later or other methods for store them, at least to transfer them from the field to the lab, but I prefer to keep them alive because they can stand alive for weeks, even in your pocket. But after that, Eva told us a lot about preparation things. You need to clean them, identify them, and then you start with the extraction protocol, which is very important at that stage. If you are intending to not just to look after CCHF or viruses, but other pathogens as well, so we probably need total nucleic acids. And because during the extraction, we crush the ticks and we've used all the material we have. So if you go with RNA extraction methods, you lose the DNA. If you go with DNA extraction methods, you lose the RNA. So it's better to use total nucleic acid extraction and to have all the genetic material. The other issue is pooling or individual testing. You can use pooling for small ticks like some Haemophilus ticks, some nymphs or larvae. But with large ticks, I prefer individual testing or perhaps maximum pools of 5. Otherwise, there is a lot of inhibition if you use more ticks. And it also affects your prevalence calculations as well. Regarding the PCR, yeah, you can start with generic nested PCR. It's better if you want to look for other pathogens and to save some money, and then go into specific level. Or you can do specific PCRs, which we are doing currently. We use qPCR to test the CCHF RNA, but I still keep the DNA and other genetic materials for testing for other pathogens. And the last thing is sequencing. It's very important to see the changes in the pathogens, or even if you go to de novo sequencing, you can find the other pathogens which you don't like, pathogenics which you don't know, but luckily you can find it. This is the tick. Can you see the video? Because
it's a bit blurry, but it's moving. Yeah.
Sorry for that. So this is this slide about some results from Turkey, And there are very different results. You know, in the past we did some nested PCR, RT-PCR, and found 16% of ticks infected. Then in the following years, despite the human cases were increasing, we found a very low prevalence in ticks by RT-PCR. But lately, in trace part of European part of Turkey, we found almost 50% of ticks infected with CCHF. Interestingly, this part of Turkey doesn't report too much CCHF cases, so it's very interesting. You have plenty of infected ticks, but no human cases. And lately, we in Central Anatolia, we did some prevalence studies with host-seeking ticks, and we did grid prevalence because it is impossible to show all the prevalence data here. And it was ranging up to 18% in 200 square kilometers area. Another thing we found is that there is a different viral load in ticks, which is very important because with the ticks with viral load, probably the cases are mild, and with the ticks with high viral load, the cases are severe. And this is the last data from prevalence. I don't, because we used the method to collect Yeloma marginatum.
That is sorry, there is no presentation. We're not sharing. It's done already. Again, sorry.
Oh no, you know. Can you see it now?
Yes.
And it's look much better.
Yeah, it looks good. It looks good. Looks good.
Okay, but we get some other ticks from the field. It was by chance, because we got some Rhipicephalus ticks and some Haemophilus ticks, which we didn't use the method to collect them, but still we had some. So prevalence in those ticks, we omit the prevalence in those ticks, and we concentrate on the prevalence of Yeloma marginatum. It still seems very low. And the other thing we found is that individual testing is better than pooling. We get much higher prevalence in ticks we tested individually. We can collect ticks from hosts as well. It's better if you go for presence or absence of Yeloma. You better check cattle first and then go for collection of unfed ticks in the ground. So we collect feeding adults from livestock and wild animals like wild boar. One important, we collect all the ticks from animals, and one important step here is to collect engorged females for later studies and keep them alive when transferring to the lab. For feeding immature ticks, we prefer hunting. There is no. Sorry.
Okay.
And for feeding immature ticks, we prefer hunting. There is, because I don't know.
Men då passar ju inte idag heller, älskling. Nej, då passar det ju inte att jag kommer idag heller.
Sorry, someone is unmuted.
Nej, inte om du ska klippa håret. Jo, jag fattar, men jag hade tänkt.
So I feel we have some technical problems. Dati, can you hear me? Tati?
Can you hear me?
Yes, sorry, there was a voice and
I
was
think
things-
it's from somewhere else.
yeah, just muted all microphone now, and you can proceed. You can proceed. Sorry.
Okay. So most important thing is to collect engorged nymphs, because you bring them to the lab and make them molt in the incubator, and then they turn to unfed adults, and then you can test them for viral presence, which is very important. If you find them positive, it's important. It's direct risk to human health. And we do the same with fed ticks, but here we have some problems with the blood in ticks, so we need to be careful with fed ticks, and because the hem is a very big issue, and we need to choose a good extraction method for that. We can do pooling with engorged ticks as well, but need to keep the number low because they are full of blood, and it can inhibit the PCR. And we do the same PCRs for those ticks as well. Can you see the presentation now?
Yes.
Okay. So in Turkey, in CCHF foci, we have cattle and to lesser degree sheep. We know that cattle are very important for maintaining the Hyaloma marginatum population. According to our calculation, one cattle can feed up to 36 female Hyaloma marginatum ticks per season. And if you do a rough calculation, it makes 252,000 larvae per cattle per season. It's a huge number. So cattle are very important in maintaining of Hyaloma marginatum population, and they have role in transferal transmission of the virus as well. Sheep, actually, sheep has a negative effect on Hyaloma marginatum population, because probably it's competing with hares for the same grass. So when sheep population is decreased, population of Hyaloma marginatum increases. We do some serology in cattle as well. Serology is very important because if you have seropositive animals, it means that you have virus in circulation, and it's probably a quite good method to predict the virus circulation. And to my experience, when you see prevalence up greater than 60%, it's great risk of human infections as well. Prevalence in the host feeding ticks is similar to prevalence in host seeking ticks. So perhaps we can use ticks from animals without going to the field to collect unfed ticks, and use them for prevalence estimation studies. It's important to see how much transferal transmission is happening in the field. We didn't manage it well, but still find if a female tick is infected, the progeny is almost always infected. So if female tick get infected, its progeny is infected for sure. The wildlife is very important, but there is not much work on wildlife in Turkey. Wild boars are very good host for Hyaloma marginatum ticks, but at the beginning of the summer, spring, after that time they start to nocturnal habitat and nocturnal activity, and it's not easy to see wild boar infested with Hyaloma marginatum after June. But before June, there are plenty infestation on wild boars. Hares are in Turkey. Hares are very important hosts for immature ticks of Hyaloma marginatum, and we found about 17% of detached ticks from Hyaloma marginatum to be infected with the virus. Partridges are very important as well. And also we found 10% of ticks from pathogens to be infected with CCHF. Well, for surveillance, not only the ecology of the host or tick is important, the socioeconomic factors are also important. So your sociology or the demography is very important. So you always need a social part of the surveillance to be done. You need to know the agriculture, landscape use, land use, and landscape of the area, because it's directly affected tick and animal ecology. In Turkey, we have undulated and fragmented landscape, and there is a lot of crop rotation, which is very good for wildlife to feed and to cattle to graze. And when the wildlife and cattle meet together, there is increase in yellow marginatum population.
Sorry, again. There is a presentation again.
Okay, it's
gone.
Yeah.
I know, worries. Maybe it could be the connection. Maybe.
Yeah, probably my connection here.
Just a question. How many slides do you have left?
Oh, 2.
Okay. No. Okay. Just wondering for the slide. Okay. Like this, it's good. When you put in full screen. No. Okay. Now it seems good again.
On the other hand, we have ecological regeneration. We have increase in bush type habitat, and it leads to increase in wildlife population. And also forestation and ban of sheep grazing is also important, which lead to yellow marginatum increase. And abandoning of the fields due to migration to the big cities is also causing the habitat ecological regeneration in Turkey. So, and as a general, after all our work in Turkey, or CCHF cases are associated with yellow marginatum presence and habitat fragmentation, and which is very good. Habitat fragmentation is very good for wildlife and livestock, and which increases the tick numbers, infected tick number. So what we need to do and what we are missing. So there is lack of rural epidemiology units in Ministry of Health.
Jati, maybe again. Also from the chat, also the others.
When I use my keyboard, it's going off. I think something weird is happening here.
And now it's good again.
Okay, there is complete lack of rural epidemiology units of Ministry of Health, which is crucial for monitoring or surveillance of any tick-borne disease. Ministry of Agriculture is doing well. They have very good system, very good local rural infrastructure, but they are not interested in tick-borne diseases. So we need some kind of good connection between Ministry of Health and Ministry of Agriculture. And there is lack of surveillance guidelines for selected ticks and tick-borne diseases. We need standardized protocol for area selection, sample size estimation, collection, transport, and identification, also pathogen detection, and tick density and infection prevalence calculations. We don't have anything about that. We need to understand the role of wildlife in circulation of the virus. We're still missing this part of the ecology. We need to understand the degree of transmission in ticks, and to understand the role of other tick species in maintaining those enzootic foci. Is there any cryptic cycles in the nature? Because we used to find a lot of other species infected, so perhaps there is some cryptic cycle going in the nature, but we see only Hyaloma marginatum because it's aggressive and it's attaching to the humans. And we need to understand the impact of climate anomalies on vector population dynamics. Okay, thank you very much.